like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. I got my head held high, I'm on my way, and I'm following the signs to my happy place. I'm walking in the light of a brand new day, and I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place. And I'm right where I belong, in my happy place.
Welcome to the National University of Singapore Yongloo Lim School of Medicine Healthy Longevity webinar. Thank you for joining us. It's a great pleasure to have you here on the show tonight, and it's dedicated to muscle and muscle research. We have Professor Nir Eynon with us. He is the expert on muscle and epigenetics, and I will introduce him in a few minutes. As always, please use the Q&A function to send your comments and your questions you have. And first, we will hear from Clarissa Lim. She is our research assistant in the Center for Health and Longevity, and she will present a recent publication about also muscle, about the impact of aging and exercise on skeletal muscle mitochondria capacity, energy metabolism, and physical function. Clarissa, the floor is yours.
Thank you very much for the introduction. Hi everyone, my name is Clarissa and I'm a research assistant at the Center for Healthy Longevity. Today, I will be presenting a paper on the impact of aging and exercise on skeletal muscle mitochondrial capacity, energy metabolism, and physical function. The paper was published in the journal Nature Communications in 2021 by Grevendong et al. At Maastricht University. One of the distinctive features in aging is the progressive loss of muscle mass and physical function. This is known as sarcopenia. The loss of skeletal muscle mass and tissue function has been related to mobility impairments, which can lead to a loss of physical independence and increased care needs. Alongside this progressive loss of muscle function, mitochondrial respiratory activity in human skeletal muscle has also been shown to decrease with age in healthy men and women. The mitochondria is an organelle within the cell, and amidst other functions, its most prominent role is to produce energy for the cell through respiration. There have been some studies that suggest a relationship between age-related changes in muscle mitochondrial function in relation to the decline in skeletal muscle function. Since there is decline in physical activity, hereafter referred to as PA with age, and PA can also affect mitochondrial function, the author set out to determine the extent to which chronological age and physical inactivity contribute to the decline in mitochondrial function and muscle health during aging. The first aim of the study was to assess if mitochondrial function is reduced in older compared to young participants with a similar level of habitual PA, and to examine how mitochondrial function relates to muscle function. To do this, the team compared young versus older individuals. The young or Y group had 17 individuals aged between 20 to 30 years old, while the older adults or O group were made up of individuals aged between 65 to 80 years old. Both groups had comparable levels of PA as measured using an accelerometer and engaged in no more than one structured exercise session per week. What they found was that the older adults, in comparison with the young, had lower endurance as seen by a lower maximal aerobic capacity or VO2 max. Furthermore, it was observed that the old group had a lower isokinetic muscle strength as compared to the young. Besides this, the group also assessed exercise efficiency and substrate utilization during a submaximal exercise test. They found that while the resting energy expenditure and respiratory exchange ratio, or RER, were comparable between old and young, both gross and net exercise efficiency were found to be lower in the older individuals as compared to the young participants. This means that the older individuals require more energy to perform the same amount of work compared to the young. There was also lower mitochondrial function in older adults, as demonstrated by the reduction in maximal FCCP-induced uncoupled respiration. They also observed that the PCR recovery rate constant was lower in older adults compared to the young. To complement this, the team also analyzed the amount of mitochondrial content, which showed that young and older adults had similar expression of the 5 oxidative phosphorylation proteins. The second aim of the study was to investigate the potential of regular exercise training in older adults for the maintenance of mitochondrial function and if this is associated with maintained muscle health. There were 3 groups. The first group was the same older adults, or O, in aim 1, trained older adults, or TO, and older adults with a low physical function, or IO. Unsurprisingly, endurance and muscle strength were the highest for TO among the 3 groups. Resting energy expenditure and RER was comparable across the 3 older groups as well. However, TO showed a higher gross and net exercise efficiency in comparison to O and IO. In terms of mitochondrial function, maximal FCCP-induced uncoupled respiratory was around 1.4-fold higher in TO than in O. Surprisingly, the PCR recovery rate constant was not significantly different among the 3 groups. Another unexpected observation was that the levels of complex 1 to 4 were higher in TO than in O. The levels of complex 2 and 4 were also higher in TO than in IO. This contrasts the results observed in aim 1, where young and older adults had no significant difference in OX4 protein levels. Lastly, they found that mitochondria and muscle function and exercise efficiency are related. In summary, the results suggest that maintaining a healthy level of PA is not sufficient to prevent an age-related decline in mitochondrial function. Nonetheless, the team was able to demonstrate in the second aim that increasing PA by exercise training can help to preserve physical function and performance. Finally, they showed that mitochondrial capacity was positively associated with exercise efficiency, further supporting the idea that mitochondria could be a promising target to negate the aging-associated decline of skeletal muscle health. That's all for me. Thank you very much for your kind attention.
Thank you, Clarissa. Very nice presentation, really showing that if you're older, you have to work a little bit harder. And maybe this is not just due to mitochondria, but maybe also to epigenetic changes. And this will lead to our speaker today, and that is Professor Nir Eynon. He is a NHMRC Investigator Fellow working in Melbourne, where I also worked at the Institute for Health and Sport of the Victoria University in Australia. And he leads the group of genetics, epigenetics, and exercise. And he has a strong relationship with NHMRC, which is the funder in Australia. He was previously the NHMRC Career Development Fellow and the Australian Research Council Fellow. And Nir has lots of other opportunities and functions too. He is also the Associate Editor of BMC Genomics and the European Journal of Sports Sciences. And he also received the Young Tall Poppy Science Award in 2000 2020, which is actually an award for the real outstanding researchers in Australia. Nir, we are very happy to have you here in the show, and the floor is yours.
Okay, great. Thank you very much for the very kind introduction, Andrea, and good evening, everyone from Melbourne, Australia. So my name is Nir Eynon, and I'm from Victoria University, and I will be speaking today on what my group is doing, which is mainly exercise, epigenetics, and aging. So my lab is very focused on or working in the intersection between epigenetics, aging, and exercise, and we're also very interested in sex differences between males and females in both exercise and aging trajectories. We have unique exercise cohorts and muscle sample biobank across lifespan, which I will talk about it later in our lab, and we also have access to more samples via our collaborators. So we're very specialized in muscle. And as I mentioned before, we are very interested in the aging trajectories in skeletal muscle. And we all know that aging is a growing health and economic burden on the society. It is accompanied by the decline in function capacity across the lifespan. And in fact, across every organ system, age stands out as the single biggest driver of disease. And as you can see here, all individuals will experience changes with chronological aging, but not all individuals are experiencing the same degree of biological aging and the same degree of predisposition for disease. So, and we also know, as discussed in length in here previously, that epigenetic changes, and it's discussed by Steve Horvath and Morgan Levine before here, that epigenetic changes are important, particularly in aging. And in particular, we're talking about DNA methylation changes that are predicting aging trajectories. And DNA methylation refers to the addition of methyl tag or methyl group on the 5th carbon of a cytosine residue that reside alongside the guanine base in the DNA. We also call them CpG sites, and they are distributed throughout the human genome at very specific locations, and they also regulate gene expression. The methylome is very dynamic and it's been changing with age. And CpG sites can be added or removed by DNA methylation enzymes, and many CpG sites have been shown to change methylation state between old and young, but also in response to environmental stimuli like exercise. And although the CpG sites are binary, it is measured over a population of cells and takes on a methylation fraction, which is a gradient between 0 to 100%. And there are ways to assess epigenetic aging. There are actually two main ways to assess epigenetic aging. One of them is the epigenetic clocks, and the other one is epigenetic wide association studies. So the epigenetic clock concept was discussed here very thoroughly by Professor Steve Horvath and Professor Morgan Levine. Steve and Morgan previously discussed that in length here, and just to recap and to remind everyone, epigenetic clocks is a machine learning algorithm that predicts the age of a sample based on DNA methylation patterns, particularly changing with age. It's also a multi-tissue, and it's pretty accurate predictor of age across most tissues, and as we heard from Steve Horvath, across many species as well. And the difference between an estimated DNA methylation age and chronological age are called age acceleration, okay? And they are biologically meaningful. So when Steve created the clock, he had very, very few muscle samples, and therefore his prediction in muscle was very poor and way off. And because my group is specialized in muscle and exercise, we embarked on that, and we actually created a muscle-specific epigenetic clock. And as I mentioned before, my group is looking at the intersection between aging and exercise in skeletal muscle. And this is a tissue that has not been previously studied in depth in relation to not DNA methylation and aging. So as I mentioned, we first developed an accurate, pretty accurate muscle-specific epigenetic clock based on the methylome of more than 1,000 healthy skeletal muscle samples from 16 independent data sets. And can I just say that this is a very big undertaking done by Sarah Voisin in my lab, and also getting muscle from human muscle with methylation data is very rare and very hard. And the first version on the left-hand side, you can see that we published that in 2020 at JCSM with just over 700 muscle samples with methylation data. And we are refining and updating the clock as we go with now more than 1,000 samples, correlation of 0.7 and median error of 4.4 years, which is pretty good for a clock and pretty accurate. And so this is considered a very accurate clock for human muscle. And as I said, we are updating the clock as we go. We also created an R package with a code in Bioconductor. And this is, of course, open access and freely for use for the scientific community. Now, we were anxiously waiting for replication because we know how important is replication in science. And in fact, last year, 2 groups used this package, use our package, and the correlation between their muscle methylome pattern to age using the MIT clock was highly significant, as you can see here on the right and on the left. And it is important to remember that this clock was trained to look at chronological aging. So it's specifically picking up CPG sites that are changing with age. And so it's not necessarily a useful tool to study the effect of exercise on epigenetic aging in muscle. And indeed, 2 recent studies that looked at exercise and epigenetic aging in both muscle and blood using the Grimm age clock from Steve Howard, the blood clock, and our MIT clock, showed no association between physical activity and the clocks. This is the Grimm age clock, and there was no association with physical activity. And in the second paper as well, no association between physical activity or voluntary physical activity, and neither with the Grimm age or the MIT clock. That is also reiterating that the clocks are not an optimal model to assess DNA methylation changes in response to exercise. So exercise has no effect on epigenetic clock in muscle. So our group has sought out to answer the following questions using a different method called epigenetic wide association studies approach, which I will talk about it in a minute. And to answer these questions, we need very well designed human studies. And the GeneSmart cohort in my lab is as such. And the questions that we ask is, first of all, do fitter individuals display younger methylation and also transcriptome? Remember that DNA methylation is affecting significantly the gene expression. So do fitter individuals display younger methylation and transcriptome profile than less fit individuals, human muscle at baseline? We also wanted to look at exercise training, controlled exercise training, and does it mitigate or reverse these omic aging in human muscle? And we also use the force immobilization study or muscle disuse approach to see if it accelerates or causes omic aging in human muscle. So the GeneSmart study or the GeneSmart cohort, in the GeneSmart cohort, we are collecting muscle biopsies at rest and in a controlled environment, including controlled diet. We then usually training those participants for 4 to 12 weeks, and again, collecting muscle biopsies at several time points, either after 4 or 12 weeks. And we also doing a lot of phenotyping before and after. So we are conducting exercise testing before and after, and a battery of tests to collect their exercise phenotypes. And as I mentioned before, we cannot assess the effect of exercise on epigenetic clock. So another way to look at epigenetic and aging is EWAS, Epigenetic Wide Association Study. So we first, before looking at exercise, we characterize the DNA, transcriptome, and proteome of human aging muscle. We use meta-analysis effect. This is on the x-axis, and meta-analysis significant on the y-axis for more than 650,000 CPG sites across the genome. And as you can see on the left-hand side, in the blue dots, this is the sites, the DNA methylation sites that were hypomethylated or decreased their methylation with age. Whereas on the right-hand side, in the red, you can see the dots that are hypermethylated, that represent differentially methylated positions, DMPs, at a false discovery rate, FDR, of 0.005, so quite stringent. So what we found, and you can see it here, we found a profound DNA methylation changes with aging. So a widespread methylome changes associated with age in muscle, more than 40,000 DMPs, differentially methylated positions across the human genome, were changed with age. The next thing for us was to integrate them with aging-associated genes and proteins available in either the literature or from our own cohort, from our own lab. When we done that, we found 57 genes in this intersection, and among them, many genes associated with muscle structure and muscle metabolism, as expected. The next stage for us was to build an interactive website called MetaMet, in which we can, or each of us can put CPG of interest or the methylation site of interest and see how it changes the genes and proteins annotated to these specific CPG sites. And this is here, the MetaMet, and it's of course freely available, and it's based on 908 muscle samples, which again, I reiterate that it's quite a big number for muscle samples. Anyone who is working with human muscle knows that typical exercise studies or even typical biopsy studies are maybe 10, sometimes 20 or 30 people. And to collect so many samples is a really big job. So, coming back to the exercise and asking ourselves, does regular exercise training is associated with DNA methylation aging in human skeletal muscle? Again, using EWAS. So after we identify the genes and CPG sites that are associated with aging, the next step for us was to identify omic data sets that have physical activity or fitness phenotypes, or have phenotypes and DNA methylation data after or following exercise training or inactivity. We did it using cross-sectional observation studies and longitudinal interventional studies. We used open access repositories, our own lab data and our collaborators' data. Overall, you can see here the DNA methylation and the RNA samples or data that we collected in skeletal muscle from the different cohorts. And overall, we reached more than 520 DNA methylation samples data in human skeletal muscle with exercise phenotypes, and almost 1,500 transcriptome data, which is quite significant. We then performed meta-analysis in DNA methylation and transcriptome, and we didn't look at proteins. And the only reason we didn't look at proteins in relation to exercise is just because we didn't have enough data on that. So it's very rare. So can I just say that anyone here has proteomic data in human muscle with exercise or exercise phenotypes, please contact us. We are very happy to collaborate. So then we tested the association between the age-related CpG sites and RNA and fitness, exercise, and inactivity in each individual study. As I said before, we then meta-analyzed the data of age, fitness, exercise, and inactivity all together. So what are the results? As you can see here on the left-hand side and on the right-hand side, this is the methylome and the transcriptome data. And on the left, we can see clear negative correlation between DNA methylation changes with age and DNA methylation patterns or changes with exercise. So this is one CpG site that you can see here. And this CpG site, or this gene on the right with the transcriptome, is changing the methylation status with age and with increase in VO2 max. So they are decreasing their methylation with age, but increasing their methylation with VO2 max. So contradicting effect. And as we can see here, we saw negative correlation with those specific sites in red that are very significant in FDR of 0.05. And again, the same pattern goes to the transcriptome on the right-hand side, VO2 max versus aging, and negative correlation. So I just want to point out that first of all, we had in the methylome, we had 3 independent cohorts with N of 355, and we had 4 independent cohorts with more than 400 samples at baseline in the transcriptome level. I also want to point out that for every increase in 1 unit of VO2 max on the left, or aerobic fitness on the left, it was equivalent to a reduction of 2 to 3 years of age. This is, of course, doesn't mean that if you increase 1 milliliter and minute kilogram of VO2 max, you will gain 3 years, because it's not across the whole CpG sites in the genome. But I should state that it does not happen in the whole genome, and only in some CpG sites that are highly reflecting the contradicting effect between VO2 max and aging. So it's a good sign. What happened with controlled exercise training? So in the methylome level, we didn't quite reach the significant level because we're underpowered. We think that we're underpowered because we had a few very different cohorts. Same thing with age and after training. You can see that there was a big negative slope, but we didn't quite reach significance after training with 4 independent cohorts. However, when we looked at the transcriptome after training, we did see a widespread association or negative association between exercise and aging in the DNA methylation level, as we have noticed before. What happened when we looked at muscle disuse, which is, if you'd like, muscle disuse, which is, I guess, a positive correlation, should be a positive correlation with aging. So muscle disuse accelerate transcriptome in aging. Unfortunately, we didn't have methylation data. So when we looked at muscle disuse, again, the model that promotes aging and physiologically is the opposite from exercise. So although we didn't have the DNA methylation data, we showed a positive correlation here between aging-related CPG sites and inactivity. So they are going in the same direction of aging. So inactivity and aging, CPG sites are going in the same direction. This is only with 170 participants. And you can imagine how hard it is to do studies on muscle disuse or immobilization in humans. So, and although we only had 170 participants, we found very striking effect of positive association between age and inactivity. So if I can summarize in this beautiful heat map, in this data, you can see that each row is a CPG site here. Each row is a CPG site. And in blue, CPG sites are CPGs that are, or methylation sites that are increased with fitness or following exercise. And the effect is, and then you have VO2 max that is increased and training in red. And you can see that the effect is very consistent across the epigenome and contradicting between age, VO2 max and training at many CPG sites across the genome. And the same goes to VO2 max training that are going in the same direction, and where age and muscle disuse CPG associated sites are going in different direction to those. So this is a very clear picture for us that showing us that the effect is very consistent across the epigenome, and it's actually the reverse or negative between exercise and aging at the epigenetic level. Okay, so I want to talk a little bit about our future direction. So what we are currently doing or are about to embark on. So as I mentioned before, the cohorts are very difficult to get. So if anyone has human cohorts with biopsies and exercise, please contact us. I'm saying that again. And we are now performing integration to pinpoint the top genes. So this is the first thing that Sarah is performing now, integration between RNA and CPG sites. The second thing is, this what we're hoping to do is to perform some functional validation, such as using a vector to methylate the promoter of some genes, and to see to look at the gene expression, if it's increasing or decreasing the gene expression. And again, one of the difficulties we have is that we are not aware of cell culture model that can mimic long-term exercise, not just one bout of exercise, which is something that is available. So it's very hard to mimic exercise in cell culture. Again, a long-term, like chronic exercise. And another avenue that we are now exploring is viable methylation with age. So this is Kristin Seal from my lab, and she's looking at big project on VMPs, viable methylation positions. So we know that VMPs are changes in mean methylation, and most of the methylation studies, epigenetic studies, are looking at DMPs, and they are changing in the mean with age. However, VMPs, which we think that has more meaning for biological aging, are changing in viable with age, meaning that the variants increase or decrease with age. And to identify VMPs, we look at behavior of those residuals here around the slope. You can see here, okay, and the change with aging. And we think that VMPs, which we currently understand very little about them, are more reflective of biological aging, but this is remain to be seen in Kristen's work. Okay, so to summarize my talk today, our data show that age has a profound effect on skeletal muscle epigenome. Individuals with higher fitness at baseline have younger DNA methylation and transcriptome profile. Exercise training, controlled exercise training, shift DNA methylation profile toward that of a younger individual and also the transcriptome. We also identified some of the top genes associated with aging and exercise in muscle. And we are now validating some of these genes using methylation vectors and also siRNA. So with that, I would like to thank my wonderful research group at Victoria University, as well as many of my collaborators, and of course the funding bodies in Australia for supporting our research by grants and fellowship. Thank you so much.
Thank you so much, Nir. Wonderful talk. I have lots of questions for you. But maybe we can start with your first slides where you showed, I think, the Howard clocks and the Levine clocks, which I think are based and standardized on chronological age, because that's how they their algorithm works. Yeah. Can you tell me, and maybe also the audience also struggling with this, if we are talking about biological age, why would you have as the standard chronological age? Why do we want to predict chronological age while looking at biological age?
Okay, that's really interesting question. And I think Steve Howard has spoken about that. And in the review in 2019, they indicated that currently the indication of a chronological age is, as I see it, is very important for things like forensic, for instance, can be important for that, but no one really is sure if there is biological meaningful for those specific clocks. I can tell you for almost for certain, or from the data that we have, that those clocks, especially in skeletal muscle, are not reflecting of exercise stimuli and biology.
Yeah, I think you really nicely showed it, that there's no association with physical activity, and there was maybe also sedentary behavior, which we would expect. Yeah. And that was the first slides. I was like, oh, why won't you associate your methylation changes with function, which you actually did very, very nicely. And I think you are the only group on Earth who really showed very nicely that it is associated with age and VO2 max, etc. So once again, congratulations.
Yeah, thank you.
That you actually are able to do that. And while thinking about the VO2 max, that's more cardiovascular fitness. And then I thought, okay, what kind of exercise should you actually do to improve that? So I would like to challenge you a little bit to maybe elaborate if resistance exercise training or cardiovascular fitness is the driving factor for your epigenetic changes you see in muscle.
So it's a really interesting question. So first of all, when we use the meta-analysis in exercise, we looked at VO2 max at baseline, and we also looked at VO2 max after training, but we also included resistant training in our analysis, and we could meta-analyze that, and we can see that any exercise together is changing the methylome or contradicting the aging methylome, which is, I think, really nice. It is very clear to me that resistant exercise is very different from aerobic exercise, and we know that. And we are currently thinking about a project with skeletal muscle and resistant training, which is really interesting.
Yes, because I think the question of at least from myself, but also others maybe in the audience, would be, okay, what kind of exercise should I do to make my muscle, at least on epigenetic level, to make that younger? And
so what we see in our data is that we're applying high intensity interval training. It's very controlled. So we are, increasing the progression every training, and we are doing it in a very controlled environment in the lab. The high intensity training seems to be working really well, even after 4 weeks, in particular after 12 weeks, but we see really striking results after 12 weeks, even in trained individuals, which is really nice, I think. So I think for me, this is a good model to work with, but it's inevitable question to ask, okay, what about this exercise? What about that exercise? I think that strength-based exercise will change the epigenome as well. And I think there was a few papers on that with very small sample size, but few good papers from Adam Sharpless group, which we are working very closely with. So I think at the end of the day, you need a lot of samples to look at the epigenome, and you need very big undertaking to combine everything together. And somehow in this big undertaking, sometimes you do lose the phenotype a bit. And I'm the first to admit that.
Yes, and then it comes up in my mind, what kind of phenotype do you then measure, especially if you do resistance exercise training and the satellite cells are really replicating and building muscle, and you increase muscle mass, not only that the fibers themselves are growing, but will that change the epigenetic phenotype because of a shift in the cellular phenotype?
That's a question that I'm asking myself every day, because we know that skeletal muscle is not the whole muscle, is, has fiber types and satellite cells and endothelial cells and others. And we are just discussing that almost every day. In fact, my group discussed that today. How do we actually, separate those fibers and those cell types and account for those factors? It is extremely hard, because even if we are looking at single fiber, for instance, type 1, type 2, type 2X, then even if we do that, and it's a very, very big job to do DNA methylation in each one of them, and actually one of my students is doing that at the moment with 15 individuals, Andrew Palmer. Even if we do that, we still have another 30 to 40% of other cells that we need to extract DNA from and do DNA methylation on and separate them. It's very hard. So I don't have an accurate question on that, on the cell type.
Maybe in 1 or 2 years, we will invite you again with the answer. But we, from the sarcopenia field, we know that we need resistance exercise training, a little bit of aerobic exercise too, but also we need nutrition. So have you seen, or have you analyzed any nutritional influences in your bigger cohort studies, how nutrition might influence certain epigenetic phenotypes in muscle?
Yeah, so unfortunately, we haven't done anything in nutritional intervention in our cohort in skeletal muscle, but I am aware of papers that looked at nutritional intervention in the context of epigenetic clock, and they are very shifting or changing the epigenome in a really, really good shape towards a healthy profile. So nutrition is also very interesting. I haven't seen paper in skeletal muscle, only I think in blood. It's really interesting, but then you need another intervention for that and a lot of samples, and that's another challenge that we have.
The last question before going to Xiaoping Guan, who has, I think, lots of questions of the audience, is you have epigenetic data, but you also link it, which I very like, to proteomics data, for example.
Yeah.
Do you think that in the end, we will more focus on readouts, more on the proteomics or transcriptomics level than looking at the epigenetic phenotype?
Wow, it's a really good question. which I don't have a good answer for. But the only thing that I can tell you is that currently DNA methylation is almost the molecule that is predicting age very well, at least chronological age, but also we see the biological aging, and we see that the changes in DNA methylation and transcriptome are very striking. So is it the DNA methylation that is driving that, the transcriptome, or the end product, the proteome? I don't know. I think we'll still use those integration because we need to know where it's coming from.
Yeah, absolutely. I agree, but I thought it would be easier. Sure thing. Tell us, what is the audience saying and asking Professor Eynon?
Thank you so much, Andrea. Prof Eynon, thank you so much for your talk. It was really inspiring and interesting. We do have a variety of questions from the audience. Maybe we start from these most upvoted questions. What are the major molecular mechanisms responsible for epigenetic aging in skeletal muscle in human? So what is the major molecular mechanism?
The major molecular mechanism?
Yes.
This is exactly what we are doing now, and this is exactly what we are undertaking now. And I still don't have the question for that, unfortunately, in skeletal muscle, because we really need to dig into those specific genes and proteins that we identified and start working on them very deeply. So we do have some proteins and genes that are off target. Some of them like HDAC4 and PFK beta and others we are now targeting and trying to see what is really the mechanism behind them.
Oh, thank you so much. HDAC4 is involved. That is very interesting.
Yeah, HDAC is a top gene in our list, definitely.
Wow. Yes, definitely. You'll find something there. And the second question is there a gender difference with regard to epigenetic aging in skeletal muscles in human?
So that's another. So that's it looks like you're just asking all the questions that are my future project or my current project, which is great actually, because it aligns really well with what we are doing, and it's a good insurance for us to show that we are going in the right direction. So we have another student in my lab with my collaborator Severin Lamon in Deakin. Her name is Mandy, and she is now embarking on a project that is looking at sex differences in DNA methylation aging in many tissues, but among them are skeletal muscle as well. So I'm pretty sure that there will be differences, but I don't have the data to show that yet.
Mm-hmm.
I just want to point out actually that we do have data, and we published data last year in Clinical Epigenetics. This is Shani London from my lab, and she published data on exercise and epigenetics regardless of aging, on striking changes or differences between males and females in epigenetic in response to training and at baseline. So we are hopeful that with aging, those changes will remain and will be seen.
Mm-hmm. That made me want to start exercise immediately. And there's another question. Currently, how close do you think we are in finding a practical and realistic therapeutic treatment in preclinical or clinical studies that can be used to improve epigenetic aging?
Wow, that's a big question. I think there is a big company now, or a few companies that are working on that. In particular, the company that is working with Steve Horvath and Morgan Levine, the new aging company. I'm not aware of the progress, to be honest. I don't think we are very close. I think we still have a way to go to do that, because we need to map those epigenetic changes across all the tissues. And I think what we need to do is to try and target epigenetic changes that are cross tissues. And this is exactly what Christine in my lab is doing now. she's looking at multi-tissue or across all the changes of epigenetic aging across many tissues like blood, liver, breast, muscle. And if we can find those CPG sites that are, shared between all those tissues, that will get us very close to maybe a better answer here.
Wow, I really waiting for the result to come
May I
out.
ask a follow-up question here?
Mhm.
Is it then, would you rather invest in physical exercise and nutrition or in, for example, medication?
I always bark for exercise. I'm an exercise person. I think exercise is important. It's important for everyone. I also think that there's almost no such a thing as someone who is not responding to exercise. I think everyone is responding to exercise depending on the phenotype that you're looking at. So you might not see changes in VO2 max in one person, but he will change, or they will change their, something else in their body, their heart or their legs or whatever other phenotypes that you measure. It's phenotype dependent. But it's just a way for us to prove and to get a very, very strong message what's happening with exercise. Look, the data that we show with skeletal muscle, that contradicting effect of epigenetic aging and exercise, just showing us that if we do exercise, we can reverse those changes. We can almost reverse some of those changes. It doesn't mean that we can reverse aging. We can slow it down, I think, by doing this exercise and by looking at those models.
Yeah, and maybe even we can have the blood samples as a readout for the epigenetic age at the end, if there's lots of overlap with muscle tissue.
Exactly. So this is one of our goals at the end of the day, to develop a simple test that you can look at CPG site or certain CPG site in blood, which is easy measure. But this is where we need to do the project, the big project with the cross-tissue and multi-tissue that is being undertaken now.
Yeah, shopping over to you again if you're not yet exercising.
Yes, and there's a type of question from the audience who asks, like, is there a optimal level of exercise? Is it like the more the better, and is it like it's optimal for different age? Like the older you don't do that much. Like, is there a correlation?
Yeah, that's a $1 million question that we're always asking. What is the best exercise and how much exercise can you do? I think you have to listen to your trainer and you have to listen to your body. I don't have a clear answer about how much exercise do you need to do at certain day. There are guidelines for the ACSM and other bodies that are giving you guidelines, but these are just guidelines. I'm doing exercise every day. There are some people that are doing three times a week. Which exercise should we do? We also showed, and other people showed many times, that high intensity exercise is very beneficial, even for elderly adults, if you do it safely, and it's quite safe and good. Sometimes it's also time consuming, but also it stimulates the muscle a lot. We also know that we need strength training when we're aging, and protein supplements. So it's probably all together.
I see. So the more muscle we have, the better, right?
Yeah, we know that already, I think.
Okay, so there's plenty of other questions. So let me ask another question. There's special interest in that people ask, do we know which skeletal muscle is first to undergo epigenetic aging?
Wow. The answer is no. We are taking muscle biopsies from the quadriceps. Mm-hmm. This is the muscle that we are taking because most human studies are taking from the quadriceps. It's the most feasible place to take. And this quadriceps has fiber type. So I can't tell you for sure that other muscles will respond differently or better. But I think it's a really good representative of what's happening in the body because it's a really big muscle.
Then, can you comment on any racial difference, like between the black and white or Asian is there any difference in their epigenetic profiles in terms of exercise?
Yeah, so I can't comment on that because I don't have the data for that, because one of the problems that we have in science is that, and not only in epigenetics, also in particular in genomics, for instance, is that most of the data is coming from, unfortunately, Caucasians and European samples. And there's very few data from places like Singapore, for instance, in Asia and Africa. And this is something that we are thinking of doing now, looking at, other cohorts, not only Caucasian cohorts. And anyone who has muscle samples from those cohorts, we are very, very happy to collaborate and talk about projects.
Thank you for reminding us, Nir, that we have to actually get it through ethics. Yes.
Exactly. Yeah, I'm talking to you, Andrea. Yes.
Yeah. One more question.
Okay, so how about excessive exercise? Is that doing more harm than good?
Well, depending on what is excessive exercise and what do you like, you need a recovery period. We all know that. You need to listen to the guidelines. You need to listen to your body. I wouldn't do exercise 10 hours a day. There are clear guidelines for that. In particular, when you're old, I wouldn't do very excessive exercise because it's creating more damage than good, I think.
Thank you so much.
Perfect. Thank you so much. I think Shopping is already exercising. I will also exercise afterwards. I think everybody is really now has a feeling, okay, let's make our muscle younger epigenetically, and that will also lead to hopefully proteomic changes and other translational changes. And it's good for your cognition and other things too. So thank you very much, Nir, for this wonderful presentation. And we are looking forward to your next results and to your next publications, absolutely. And we're looking forward to collaborate with you.
Thanks for having me here today.
For the audience, please use the chat function and leave us your comments and feedback, and the panelists and all attendees option. We also have some exciting news from our center and our School of Medicine, the Yong Lin School of Medicine, after the end credits. And we also have a community webinar next Saturday. There's a poster after the end credits, and it will also be in the chat. So if you would like to participate, please register. And it's about muscle. On March the 17th, Brian Kennedy will be the host, and we'll have Professor Vera Gorbunova with him. I can tell you this is wonderful research. And we will end this nice talk, thanks again, Nir, with a video, which is also about muscle. And we talked about workout. And I would say, please have a look at this video, how you should work out when you are 100 plus years old. Take care.
It's kind of a question of health. Nature will tell me when it's time to stop. But if I'm healthy, I'll go on, of course. People have said, that's what you said to me. Surely, John, when you're 100, you will watch. Well, I tell you what I'm driven. I've been driven all my life to try to, I'm always about competing with myself. Try something a bit, go harder. See if you can do it, that's the attitude. And I think the result of that, here I am, who knows. I always had this feeling I could do a bit more, and I think that's the way to do it, really. Finish, not strain. There's no strain in what I do. I try and keep within my I challenge myself when it comes to weights. I'd like to go a little bit heavier. If I feel like sleeping, yes, I would do. But I don't feel like it. I leave here energized. I leave here with my body feeling Vitality is there, and my mind is clear. Good, you know.
I'm feeling like sunshine, like springtime, like something's in the water and I'm taking a deep dive. I'm feeling so weightless, like I'm gonna make it, and nothing in the universe can take this. I can see it clearly now, nothing gonna bring me down.